Remember when I mentioned to you about how the neurotransmitters effect can also be affected by the types of the receptors. Okay, so the receptors on the postsynaptic neuron can be either ionotropic or metabotropic. So as the name suggests, let's look at the ionotropic first. The ionotropic receptor, it means that the receptor itself is also an ion channel. Okay, it is an ion channel. So for example, therefore it produces a direct action. So when the neurotransmitter binds to the receptor, okay, it then causes this ion channel to open and allows the ions to flow into the cells. This receptor is also known as chemical gated ion channel. Why chemical gated? Remember when we talk about the chemical synapse, we talk about the VGCC, voltage gated calcium channel. This calcium channel only opens when there is changes in the voltage or the membrane potential of the neuron. But for the chemical gated ion channel, it opens when there is a chemical that binds to the receptor. In this case, the neurotransmitter that binds to the receptors. Because it allows a direct influx of the ions into the cell, it doesn't involve any second messenger to open the ion channel, and it produces rapid. However, it is quite short acting in terms of the response. The other type of neurotransmitter receptor is known as the metabotropic receptor. As the name suggests, it involves some metabolic processes or metabolomic as the second messenger. So it has an indirect action, meaning that when the neurotransmitter binds to the receptor, what happens is that it can activate the G protein, which then activates the adenylyl cyclase and several other downstream mechanisms, which then eventually open the ion channels. Therefore, it has a much slower effect because it needs to activate the cascade of proteins after the G protein. However, it has a more long-acting action because you can imagine, for example, this G protein, it can activate a number of other proteins, which will then probably open a lot more of the ion channel. Now, we talked a little bit about excitatory and inhibitory neurotransmitter. So let's talk about the excitatory and inhibitory synapses. Now, as the name suggests, usually in excitatory synapse, you will have the excitatory neurotransmitter, whereas inhibitory synapse will involve the inhibitory neurotransmitter. We discussed this before. Excitatory cause depolarization, inhibitory cause hyperpolarization, and eventually the changes in the membrane potential in the postsynaptic neuron, which is a graded potential, is known as the excitatory postsynaptic potential, EPSP, or inhibitory postsynaptic potential, IPSP. In the excitatory synapse, because the membrane potential is depolarized, it is closer to threshold. It is about -55, which is at which the neurons can fire AP. Whereas for inhibitory synapse, the membrane potential is more negative, therefore it is further from the threshold. So what causes the depolarization? Okay, becoming less negative. Okay, think about it. First, of course, there's sodium influx. How does the sodium comes in? When it's open, sodium channel. Or it can also reduce potassium outflux. So you haven't learned this, but let me tell you, in the cell, the main cation in the cell is potassium, whereas the main cation in the outside the cell is the sodium, whereas the main anion outside the cell is chloride. Okay, so you have a lot of sodium, a lot of chloride outside the cell, and a lot of potassium inside the cell. Now, because of the concentration gradient, the potassium tends to move upwards. But if we reduce the potassium outflux, then the potassium stays in the cell. Therefore, the cells become less negative as well. And similar to the chloride will tend to move into the cell down the concentration gradient. But if you can reduce the potassium chloride influx, then of course the inside of the cell become more less positive, less negative as well. Another way is that you can reduce the excitatory receptor on the postsynaptic neuron or decrease the inhibitory receptor on the postsynaptic neuron. So there is this. So in conclusion, you can use this mnemonic leap. When there is membrane potential is less negative, it produces EPSP, which then fire the action potential. How do you hyperpolarize a cell? Okay, in an inhibitory synapse. So remember, when the cell to becomes more negative, so we want more chloride ion. So of course you want more chloride influx. So opening of the chloride channels, and we also we do not want the positive ion, so we want more potassium to go outside the cell. Therefore, we can increase the potassium outflux, and of course, we want to also to increase the inhibitory receptor and decrease the excitatory receptor on the postsynaptic neuron. So in conclusion, it is when there's more negative membrane potential, it causes IPSP, and the action potential is not likely this time. Now, remember when we talk about the synaptic arrangement, many to one. So there's many, which will then lead to integration of the information. Now, integration of information can either be temporal or spatial. Okay, let's look at the temporal summation first. The temporal summation means that this neuron receives a lot of information in a very small amount of time, and usually that information comes from one neuron. So the inputs come from the same presynaptic neuron in succession, and this depends on the time constant. What does it mean by time constant? It means that how fast can this action potential dies out. For example, action potential 1 dies out in 5 seconds, whereas action potential 2 dies out in 10 seconds. Therefore, the time constant of this one is longer compared to this one. Okay, let's have a look here. If this action potential, for example, this action potential has a longer time constant, and suddenly midway before it dies out, the second action potential arrive, what happens is that it will submit on each other. It will be overlapping, and therefore it may produce a higher membrane a higher, it can produce action potential. But if the action potential has shorter time constant, and whenever this has already dies out and only the second action potential arrive, it won't overlap with each other. Therefore, it does not cause summation. Now, spatial summation is another type of summation. So this time, the presynaptic inputs comes from a number of presynaptic neurons. So this is what we call as many to one, right? So this postsynaptic neuron will have to integrate all this information. Now, this will depend on the length constant of that neuron itself, meaning that how far down the axon, down the neuron does the action potential dies out before it meets there's another action potential. Now, for example, this action potential, okay, for it to die, it only dies out when it's about 1 millimeter. When it travels down 1 millimeter, then it dies out. Therefore, at about 0.4 millimeter, when there's another action potential coming, this action potential will overlap with the first action potential, and it causes summation. However, if the short length constant, if the action potential has a short length constant, for example within 0.2 mm, it already dies out. Whenever there's a new action potential coming at 0.4 mm, then it does not submit. so it won't summate with each other. Now with spatial summation, because there is different input from different type of cell, this could be excitatory, this could be excitatory, but this could be inhibitory. So what happens is that it needs to add up all the input. For example, this one cause depolarization, this one cause depolarization, but this one cause hyperpolarization. You can imagine if there is EPSP and EPSP, it causes greater depolarization, right? But if there is EPSP and IPSP being received by the same neuron, it may cancel out each other effect. Therefore, there won't be any changes in the membrane potential. So this is just another example, not another example, a diagram that summarizes what we talked earlier. Inhibition can occur if the presynaptic neuron is a release inhibitory neurotransmitter. Okay, but it can also the inhibition can also occur at the postsynaptic neuron. Okay, how does that occurs? So the presynaptic inhibition is when there is inhibition to the presynaptic neuron. So for example, this presynaptic neuron is excitatory, but then there is this presynaptic interneuron that synapse on the excitatory presynaptic neuron. Or it can be postsynaptic inhibition, meaning that although this is excitatory and it is not being inhibited, suddenly the interneuron here is an inhibitory interneuron, and although this one is excitatory, it actually excites the inhibitory interneuron, therefore producing hyperpolarization of this postsynaptic neuron. So this is the postsynaptic neuron.